In the first lecture of our exploration into metabolism, we're going to start off with the first portion of carbohydrate metabolism. We'll start off by talking about how glucose is transported through the cell. There's GLUT 2 and 4. 2 is found in the liver and pancreatic beta islets, and it has a high Km. It's a low affinity transporter in hepatocytes and pancreatic cells, and after a meal, blood traveling through the hepatic portal vein from the intestine is rich in glucose. This is where GLUT2 captures the excess glucose primarily for storage. When the glucose concentration drops below the Km for the transporter, much of the remainder bypasses the liver and enters the peripheral circulation. Because of this, the Km of GLUT is quite high. GLUT4 is in adipose tissue and muscle and responds to the glucose concentration in peripheral blood. The rate of glucose transport in these two tissues is increased by insulin, which stimulates the movement of additional GLUT4 transporters to the membrane by a mechanism involving exocytosis. The Km of GLUT4 is close to the normal glucose levels in blood. This means that the transporter is saturated when blood glucose levels are just a bit higher than normal. Because of this, it's noted as having a low Km. The first part of carbohydrate metabolism is known as glycolysis. This is not requiring oxygen, and the products are NADH, ATP, and pyruvate. It simply converts one glucose into two pyruvate molecules. NADH is also produced and is oxidized by the electron transport chain in the presence of oxygen, or by cytoplasmic lactate dehydrogenase when oxygen is not present. Before I go into carbohydrate metabolism proper, take note that there's a lot of products and enzymes and intermediates. If you can memorize these, great, but by and large for the MCAT, they're more concerned about the end results in products, less so the intermediates. Other monosaccharides used include galactose, which is derived from lactose in milk, fructose, and sucrose. Both enter the metabolic cycle in a different way than glucose, though. There are a few relevant enzymatic steps when we look at glycolysis. Glucokinase and hexokinase, PFK1 and pyruvate kinase. These are irreversible, meaning that once the reaction goes past this point, it can no longer revert. Pyruvate dehydrogenase converts pyruvate to acetyl coenzyme A. This is also stimulated by insulin and inhibited by a feedback inhibition by acetyl-CoA, which is its own product. This is the precursor for the citric acid cycle, but again, without the presence of oxygen, the citric acid cycle will not proceed and thus lead to fermentation. Here's a summary of the regulation of glycolysis. Again, there's a lot of intermediates and a lot of steps, but a general overview will often suffice when you're taking the MCAT. Glycogenesis and glycogenolysis are two processes other than glycolysis that the body uses to retain its metabolism. Glycogen is a branch polymer of glucose and it's used as a storage form. The synthesis and degradation occurs mainly in the liver and the skeletal muscles, and there is some in other areas, but these are the primary two locations. Glycogenesis is glycogen synthesis, or, genesis, glyco, so glyco, beginning. This occurs when excess glucose is present, more than the body can use. And glycogenolysis is the breakdown of said glycogen. Lysis, right, is the breakdown. And in fasting states when glucose is more scarce, it's going to undergo this process to break down glycogen as an energy source. Gluconeogenesis occurs in the cytoplasm and the mitochondria, and it's mostly done in the liver to regulate glucose levels. Glucagon and epinephrine will promote this, and then it will be inhibited by insulin and excess glucose via negative feedback inhibition. It's the opposite of glycolysis. It'll break down glycogen stores to make more glucose, and it occurs during a fasting state when glucose is scarce as well. The pentose phosphate pathway, also called the hexose monophosphate shunt, is another process that produces NADPH. It also creates ribose 5-phosphate for nucleotide synthesis later on. The rate-limiting enzyme is glucose 6-phosphate dehydrogenase, and NAD+ is going to activate it, whereas NADPH and insulin are going to inhibit it. Again, NADPH is a product, so this is a negative feedback inhibition. NADPH is important in biosynthesis, especially fatty acids and cholesterols. It's also bactericidal, and it protects against reactive oxygen species via antioxidant mechanisms. Here's a quick overview of glycolysis. What I want you to look at is the summary at the bottom. You'll note that one glucose goes in with 2 NAD+, 2 ADP, and 2 phosphate. leading to 2 pyruvate, 2 NADH, and 2 ATP. That's the most important part to take away from glycolysis. Obviously, knowing all the steps is going to be very helpful, but overall, if you know the products and where these will go, that's often going to be good enough. In the next lecture, we'll finish up our discussion of carbohydrate metabolism by going into the aerobic portion of it.